Okay, Tommy, it's great to see you again to the second part of our podcast, which is going to be very interesting, I think, with very nice examples of discovery efforts and approved drugs that are really outstanding. All of them, obviously, peptides or derivatives of peptides. So please, it's a pleasure to see you again.
And a pleasure to see you too, Immanuel, and everyone out there that may be listening to this webinar. This time, I wanted to kind of focus on intracellular targets and the peptide design strategies and concepts, and some highlighted examples of the whole field that has been evolving, probably now dating back to the 1980s, in terms of identifying intracellular targets and then peptides from natural products, and ultimately from structures of targets that we can optimize through structure-based design and de novo methods. So in this introductory slide, which I showed last time in the context of my own career, I would just say that a large portion of my efforts over the years have been focused on intracellular targets, including proteases and kinases, as well as phosphatases and GTPases, such as KRAS, as well as transcription factors. And of course, many of these involve protein-protein interactions. And the best of it all, from my perspective, is that X-ray crystallography and more recently cryo-EM and related methods, NMR and so forth, HDX mass spec, all have benefited tremendously by going after intracellular targets because the availability of the protein and being able to crystallize and achieve in forms that are amenable to structural biology. If we look at the timetable, which I also shared last time, in the context of advancing the field through waves 1 and 2 for extracellular and receptor targets, and then what we'll talk about today, intracellular enzyme and protein-protein interaction targets, we see that there are some very excellent examples, including cyclosporin A, reported in 1983, which we'll talk about a bit, as well as cell-penetrating peptides or CPPs, as you'll see on the screen here, identified in 1988 with the finding of HIV-TAT as a progenitor cell-penetrating peptide. Also, intracellular proteases had a major impact in the late 1980s, and with the first report in 1990 from my own labs at Upjohn Company, with the publication of the first synthetic HIV protease inhibitor, H1749, which we'll look at in a second. And then finally, I think exemplifying protein-protein interactions as intracellular targets, the work that came out in 1990 relative to sarcomology 2 antagonists, and that also stemmed from my past work, first at Park Davis and then at Ariad Pharmaceuticals, the latter which is now part of Takeda. And here we're looking at just a few target examples, including those for cyclosporin A, which involves protein-protein interactions with cyclophilin and calcineurin. And then 3 examples of protease inhibitors, including those targeting HIV protease, shown in the upper left, with the Upjohn compound, a progenitor for the field, U81749, and Roche's sequinavir, which was the first FDA-approved HIV protease inhibitor. And now, of course, the field has evolved significantly with cocktails of HIV protease inhibitors, which take into account mutations of HIV protease, which come about for any particular synthetic inhibitor that has been advanced. So cocktails work very beautifully to be able to limit such mutations in terms of their ability to keep driving the syndrome. Then two other proteases, just quickly, include proteasome, Velcade being the first approved proteasome inhibitor for cancer, and HCV protease more recently with respect to two examples, Gratroprovir being that which was developed by Merck. So protease inhibitors clearly have been excellent targets for peptomimetic approaches, and utilize significant transformational chemistries to take the peptide to a mimetic thereof.
Can I ask you a general question regarding these
Sure.
peptides? So there are a few different chemistries here, I see, from stapled alpha helices to peptidomimetics that differ in their conformation, their size, their chemistry. Generally speaking, the intracellular targets are considered more challenging for peptide-based drugs. So can you comment in general how this was tackled in the past, and maybe a comment on how do you see the future of trying to design and discover and develop intracellular active peptides?
Sure, that's actually a very timely question, because I was kind of focusing on targets and not so much the design at this point. And I think probably it's good to actually bring the chemistry in, if you will. So to be honest, I think that going back into the 80s and even into a good portion of the early 1990s, I think it's fair to say that we all were learning about how to optimize for intracellular targets in terms of permeability. Nature handed us a golden egg, so to speak, with cyclosporin A relatively large peptide. We'll look at it in a few minutes. 11 amino acids, which includes a number of N-methylations of the backbone. That, in retrospect, was a big clue to design, because it was basically saying that NHs at some point in terms of the number will begin to work against you probably in any template or scaffold from a chemistry standpoint. It's not just molecular weight, it also is the nature of hydrogen bond donors and acceptors and other hydrophilic aspects, charge and so forth, which limit cell permeability, especially with respect to passive transport. So cyclosporin was a stunning example of a molecule relatively high molecular weight, which was able to traverse membranes to achieve intracellular localization or uptake, even despite the size, because I think it had a lot to do with the backbone and methylation of a high percentage of all the amino acids. In the case of protease inhibitors, they are, generally speaking, lower molecular weight than cyclosporin. However, they also have minimized hydrogen bond donors, if you will, and they are all passive permeable. So there's different learnings that have happened, and I think that's what I would emphasize, is we're learning as we go, and until you really have crossed the finish line with a new template or a new concept for cell permeability, we don't know exactly what the rules might be. There are rules, if you will, imperfect as they may be, really, but still relatively good for passive permeability. What are the rules for other mechanisms of cell uptake, such as partitioning that does not involve passive permeability, but lipophilic partitioning? That is probably most well exemplified by the stapled helical peptides at this point, at least at this point in the presentation. And ATSP 7041 and the aileron clinical candidate 6924, now advanced to phase 2, illustrates the concept of lipophilic partitioning. We don't believe that these peptides are getting into cells by cell-penetrating peptide mechanism per se, but rather a lipophilic partitioning, so not endosomal pathway. More recent examples, which are not illustrated here, which is actually cell-penetrating peptide approaches by conjugation or hybridization of cationic partitioning into membranes. I think we will see that making significant progress, and it is, as exemplified by the company Entrada Therapeutics. Beautiful work that's been achieved there based fundamentally upon the academic work of Dewa Pei and his team at Ohio State. So relative to design, we're learning a lot based on structure of the peptide itself, based on structure of the target itself, and learning how to incorporate design modifications of the scaffolds, whether they're extended conformation, whether they're helical conformation, or whether they're beta turn type conformation. And I think all are now excellent approaches to advance intracellularly targeted peptides. Does that address your question, Immanuel?
Yes, thank you.
I think this slide kind of brings it all together in the context of protease inhibitors, HIV protease, proteasome, and HCV protease. And in all cases, we're looking at, comparatively to cyclosporin, some pretty small molecular weight-wise peptides, but all are leveraging, I would say, molecular recognition to their targets. And also, in the case of proteases, the active sites of proteases will recognize and cleave a peptide. So to speak, the nomenclature is P1 and P1 prime to be the first and second amino acids of the cleavage of a peptide substrate. And what I've highlighted here in the pinkish background, if you will, are the replacements of the amide that is otherwise cleaved or recognized by the active site of the protease by substituting that amide with a non-hydrolyzable amide bond replacement. In the case of H1749 from Upjohn and Roche's sequinavir, those modifications are hydroxyethyl or hydroxyethylamine. In the case of bortezomib or Velcade from Millennium Pharmaceuticals, a replacement of the amide and the whole C-terminal sequence that would follow it in terms of a substrate type of template was replaced by a boronic acid. In the case of gratzaprovir from Merck, to inhibit HCV protease, this unique acyl sulfonamide replacement was very effective as an active site binding element to the protease. So different features, different chemistries, but ultimately all giving us really good examples of passive permeable, orally active peptidomimetics, and all are orthosteric to the substrate, which I think is important these days because many companies are pursuing, for some targets, allosteric as well as orthosteric, especially when we're looking at intracellular protein-protein interactions, that the possibility of allosteric is something you don't want to dismiss, but actually have screening ongoing for that purpose. I would say that the next, most forthcoming examples involve protein-protein interactions, and historically, both in the far past, if you will, cyclosporin, and in the more recent past, the work from Ensemble on ZIAP BER3 protein-protein interactions really exemplifies something rather phenomenal in my perspective, and that is cyclosporin illustrates the multiple N-methylation of the backbone, which clearly has a lot to do with passive permeability. The peptide, when you look at it, also is very rich in just hydrocarbon side chains. And in recent months, actually, and as well as years. We've seen this again in terms of what has been described as de novo design cyclic peptides, including peptides originally coming from mRNA type of strategies as well, but I would say maybe more perfected to make the point that you can reproduce cyclosporin with cyclic peptide templates, especially if they do not have too many hydrophilic or ionically charged side chains. And that work has been illustrated from both Novartis and David Baker's group in terms of cyclic, very hydrocarbon modified peptides showing passive permeability. Very elegant work and has stimulated the field rather significantly. But as I always say, when the rubber hits the road, you need to bring in side chains that are going to provide you molecular recognition to your target. So oftentimes that means that you're not going to have the example of cyclosporin here to achieve that type of molecular recognition. You're going to have, histidines and arginines and glutamics and threonines and other hydrophilic side chains. So that's going to take you into new challenges on how to ultimately optimize your peptide. In the case of the ensemble, we have a molecule which turned out to be not a monomer, but a dimer after the click chemistry introduced a cyclization, which was not necessarily limited to just a intramolecular, but a cross-coupling between two monomers to make this C2-symmetric dimer. And as we look at it, it does include two proline modifications. So you have N-methylation or alkylation in the context of proline, and also the N-terminus of each monomer has N-methylation of alanine. But on the other hand, it contrasts rather significantly to that of cyclosporin. And I think basically it just says that we learn as we go, we get surprises along the way, but we need to take into account that sometimes they're not just exceptions to the rule, they're just giving us a first insight to new rules. And so I think that there's a translocation component to perhaps the ensemble peptide in addition and beyond to that of passive permeability. Yeah. And this is important thing in terms of not just getting into the cell, but also oral bioavailability.
That's very interesting that such a huge difference in properties, and both of them are intracellular, like penetrating cell. The cyclosporin example, actually, I think it's probably was the biggest inspiration for trying to discover new intracellular active peptide-based drugs, and I think probably also a very strong push towards cyclic peptides in this regard.
Yes, it was. And, it was not the easiest chemistry to kind of recapitulate. Academic groups put, I would say, quite a bit of effort into it back in the 80s as soon as the structure came out. And it was determined that it was very hard to make peptides with multiple N-methyls like this. But you're exactly right. Currently, there are a number of companies that have evolved over the last 10 plus years that are doing, I would say, tremendous work to advance the design concepts behind cyclosporin. As much as I hate to pluck out of the vast sea of academic and biotech or pharma work in this area, I can say that some companies like Unnatural Products that spun out of Scott Lokey's group, and Circle Pharma is another example, likewise, taking different approaches, have advanced the knowledge that we now have with respect to smaller versions of cyclosporin, looking very promising and very exemplary to go after targets, whether they're receptor or extracellular, or maybe more importantly to the field, the vast intracellular target space. Companies such as Peptidream that have evolved mRNA display strategies to make cyclic peptides, but not in the same exact nature as cyclosporin, which is head to tail, but rather side chain to and terminal cyclization using thioether chemistries. They have clearly advanced the field against a plethora of intracellular targets with their partners, which include many across pharma and biotech industry. Collectively, the knowledge coming from all of that, if one was to be able to wrestle with access to all data from all companies working on intracellular targets using macrocyclic peptides, it would be the dream of a lifetime, I think, for from an artificial intelligence or just a database. Unfortunately, we have islands of proprietary information there, which we only have access through publication. But there's been quite a few publications now on this, and I think that it provides really a more than just grassroots, a fundamental database. Again, I would say that it's not all passive permeability, but some companies, and here's where I would just exemplify on natural products. They have a very powerful platform now and experience to look at passive permeability in the context of experimentally optimizing that permeability property. And they speak to Pampa very effectively now as a critical tool in that regard. When we talk about de novo design, we can now incorporate all these ideas, concepts into the design itself. And I think this is where Pepticom is at a cutting edge to do this, because we're building the molecules. You guys, and whatever I contribute to as an advisor, are capturing, the de novo design element of it, but also can also think about it in terms of the cell uptake mechanism and whether it's passive permeable as a driving feature, or whether it could be yet other mechanisms like lipophilic partitioning, or even potentially cationic partitioning. Anything is possible. I think the design of the molecule itself is the most critical to begin with to get a potent binder. But then we can now begin to optimize based on permeability concepts.
Yeah, another comment about cyclosporin and cyclic peptides in general. So first of all, I completely agree with what you said. Specifically in Pepticom, we have a few cyclization chemistries, and to see cyclization is one of them. And we find it very helpful not only in cell penetration, so also in other drug-like properties like stabilization and things like that. Another comment regarding cyclosporin is that the story behind its discovery is very, very interesting, with non-ribosomal peptide synthesis in bacteria, which I think is a very interesting field that is, generally ignored when you learn biochemistry. It's like a niche thing, but it's a very interesting story that I really encourage people to look for.
I agree. The field of peptide drug discovery has never been so exciting as it is now. And I think I mentioned that last time, that I'm happy personally to still be in the game and be able to look at it with a long-term perspective from the early days to where it has evolved now, especially with all the technologies that are at hand. And in terms of the technologies, I've mentioned natural products, mentioned mRNA methodologies, illustrated last time when we talked about the PCSK9 work from Merck, which was supported by Ra Pharma, which is now part of UCB, but that was mRNA-based methodology to discover the leads. I've mentioned PeptiDream today, as well as a powerhouse in this regard of using mRNA macrocyclization chemistries to discover leads. In this case here, looking at intracellular target KRAS, which is obviously at the top of many companies, both big and small's pipeline hopes and dreams, and has been successfully now developed in terms of FDA approval for small molecule inhibitors that target the G12C with warhead modified small molecules that interact with the cysteine sulfhydryl. We now have these examples of peptides as preclinical leads, just to illustrate both phage display with the Takeda work on the left and synthetic libraries using one bead, one compound type of approach from Dale Pace group at Ohio State. So two sources of diversity driving the identification of leads. In the case of Takeda, also an X-ray structure of KRPEP2D, which includes macrocyclization via disulfide, which is obviously doable with phage display, and incorporation of flanking tetraarginine substitutions, which collectively give you the ability to penetrate into cells through endocytic mechanisms, so cationic driven partitioning. That is recapitulated in the bicyclic structure shown at the right from the Pay team at Ohio State, in which they incorporate 3 arginines and a lysine, some of which are within the bicyclic structure itself, and 2 of which are exocyclic to that. So very innovative approaches from a chemical structural standpoint, very much the source of, I would say, new imaginative ways to also further cyclize, maybe to monocycles or bicycles, or who knows, maybe even tricycles if we look at the Merck work on PCSK9, and then leveraging, in that case, not just enthalpic opportunity, but entropy in terms of the design. So I think that this is really outstanding examples of looking at a very difficult and challenging target like KRAS, but taking advantage of diversity to get in the door with potentially, relatively tight binding inhibitors that are in the low nanomolar range. So this is just exemplary for the whole field, looking at other intracellular targets, whether they're enzymes like KRAS or not, protein-protein interactions, which we've talked about in the context of cyclosporin or ZIAP-BR3 that we just spoke about a bit, but also other transcription factors as illustrated, which will be the next topic of discussion here, looking at helical protein-protein interfaces. So as you said, Immanuel, very excellent work that is coming out of the literature now. And I would say that it's actually challenging not only drug discovery, but it's challenging just to keep up with the literature now. So at least speaking for myself, knowledge is at the hiatus of the field right now with respect to peptide drug discovery. And as we look at protein-protein interactions, I think it's fair to say, based on structural biology, that the type of protein-protein interactions typically are now falling into 2 big categories. One is helical interfaces, and the other is kind of beta hairpin turns. So a little bit of beta strand with a turn. This slide here just illustrates helical interfaces, and looking at cancer cell pathways, we have all the biggies accounted for here with respect to helical interfaces. They include P53, which I will talk about a little bit in context of work done at Aileron, but also in the context of other major nodes like beta-catenin, which is in the news recently from Fog Pharma, Greg Verdine's company, going after beta-catenin. There are other companies engaged in beta-catenin for certain that may or may not be taking a helical approach. We just looked at KRAS as a target also, and that can include both potentially helical approaches or not. We've seen examples which are not helical approaches, which are working perfectly fine and look to be very promising ways to optimize. We also have EIF4, Notch, BCL2 family in the lower part of the slide, and MYC, which is kind of in the middle, which is probably one of the ultimate targets for cancer, to go after MYC, MYC-Max, and so forth. And also in the context of HIF-1 alpha, MYC and HIF-1 are also looking to be very promising targets for macrocyclic approaches, including stapled helical peptides. So I think that if the question were, are these just good ideas and look good, or can they be translated into drugs of the future? And I would hesitate to say it's a work in progress, but it's a work that's making significant preclinical advancement. And in the case of the P53, we're in the clinic with aileron 6924. P53 is suppressed by two major suppressor proteins, MDM2 and MDMX. There are small molecule here exquisitely potent and selective for MDM2, not necessarily by design or hope, but are nevertheless super specific for MDM2. And they are now making significant headway relative to development as the next cancer therapeutics. P53 has a beautiful history that goes back into the cell biology of P53, with notables like Sir David Lane and others that have contributed to it, who probably, like myself, are hoping and waiting to see something materialize out the door, crossing the finish line in terms of FDA approval. And I think that day will come, hopefully sooner than later. In the case of how could we design a P53 drug, what we want to do in this case is go right after the suppressor proteins so that they're not binding up wild type P53 in the cell, but allowing it to be free, to liberate it from the suppressor proteins, then to drive the killing of the cancer cell. So we're basically letting your body work the way it was intended to, but without the impact of the negative regulatory properties of MDM2, MDMX. So we start with P53. And as you might see in the graphics inserted in the bottom right, the stapled alpha helical peptide analog of P53, the one that's kind of denoted with the arrow pointing to the very high micromolar range, it was a prototype looking at the P53 sequence itself. But after waves of effort to try to optimize that sequence, work done at Aileron while I was there as the chief scientific officer, we hit the wall. We were able to get relatively close to mid 1 to 10 micromolar range with analogs of P53 itself. But what was the breakthrough in order to achieve micromolar or submicromolar inhibitors, and those that were relatively close to the potency of small molecules such as Nutlin 3A, shown on the graphics as the kind of the blue line, what we needed to beat clearly to have something promising. And that was work done, by the way, in collaboration with Roche, who were the discoverers of the Nutlin series from Nutley, New Jersey. That's why the name came about. We did it, but it was done by going back to the drawing board, so to speak, and actually using phase display lead sequence that came from the Moffitt Center in Tampa, Florida, as a template. And that had some homology to P53 sequence, actually homology for the key residues involved with molecular recognition, but also other variances in the sequence, which made it empirically to us much more compatible to advance for cell permeability. And voila, that's what we were able to achieve with modifications that led to Aileron 6924, but in the context of the progenitor ATSP 7041 shown here on the slide. And the color code for the amino acids also correlate to the color code in the 3D structure shown in the middle bottom of the slide, relative to the green for hydrophobic, the yellow for hydrophilic, and the red for the glutamic acid negatively charged. The negative charge, I would just highlight to say it would not be the first thing that most people would do to make a cell-permeable peptide, whether it's macrocyclic or not. However, in the context of thinking differently in terms of specific elements such as charge, by looking at it as the accumulation of properties of hydrophobic surface versus hydrophilic, in the case of ATSP 7041, we actually have a lot of surface that's hydrophobic and a valley, if you will, running down the helix that's hydrophilic, including the glutamic. So I think that's an important design now relative to stapled helical peptides, is to think about them three-dimensionally in terms of hydrophobic surface versus hydrophilic, and trying to take advantage of the hydrophobic. We also were able to achieve X-ray structures of ATSP 7041 and analogs, including one for aileron 6924, which is yet to be published, but will, which all illustrates that the staple itself not only stabilizes helical conformation and the amphipathicity associated with that, as illustrated in the slide here, but also provides a little bit of molecular recognition to the binding interactions with MDM2 and MDMX. So I would say that there's probably entropic contribution relative to the monocyclic structure in the double turn context of the helix, but there's a little bit of enthalpy as well. It's probably difficult to differentiate all of that, quite honestly, when we're looking at optimizing any macrocyclic peptide. But I think these days we need to be thinking both enthalpically and entropically, because that's clearly where it all comes together on the battlefield, if you will, to lead optimization. Maybe the main thing that came out of this work was, as beautifully and graphically shown in the slide here, of looking at peptides and optimization of those to get to ATSP 7041, and then ultimately the aileron clinical candidate, which would be still lower in the right-hand corner as far as potency against tumor cells. Cell permeability was critically important. And until I actually had the opportunity to go to work to help rebuild in some ways and integrate the peptide drug discovery effort, we didn't have the ability to look at cell permeability per se, but only in the context of cell functional potency to understand cell uptake. One assay that I'm very proud to have been part of helping to create at Merck, and what is somewhat similar to that developed by Joshua Critcher at Tufts University, called the CAPA, C-A-P-A. The nanoclick assay is published last year in ACS Chemical Biology, illustrates a beautiful cell-based assay in which one can determine how well your peptide gets into cells relative to a relatively simple readout, a Brett assay, if you will, called nanoclick assay. I won't take everyone through it because you can go to the paper and appreciate the technical details of how the assay is performed, but it's very straightforward. And all one needs in this case is a azido-modified peptide, azido-alkyl modification. In the case shown here at the upper middle of the slide is a azido-lysine substitution at the N-terminus. And you're all set to go with a two-step copper-free click chemistry that drives the peptide to complex with a nanoclick or with a nanoluciferase halotag fusion protein through a constrained alkyne, which is desperately small on the screen here to see, but it is a constrained tricyclic alkyne that makes it all happen. And when we look at what we learned from it all, we can say that cyclosporin, which is inserted in the bar graph in the middle, is the winner, perhaps of no surprise. It was the winner from the day it was discovered by Novartis, and it still is a striking example of cell permeability via passive transport. But it's not alone. There are now examples, including Scott Lokey's analog, which is sort of in the middle in the lime green bar graph, the Zito-Lokey, we call it, comes right up there as number 2. But now we have many examples that illustrate cell-penetrating peptides. In fact, most of the bar graphs here exemplify cell-penetrating peptides, including the lowest bar graph, which I would just point out is the O negative example of one of the cell-penetrating peptides. Most of them are linear, which have been developed up to the days of Dewa Pei's cyclic peptides, which are exemplified here as CPP9 and CPP12, sort of in the middle right of the bar graphs. But also we bring in the hydrocarbon stapled peptides to the left furthest black bar graph. That is the stapled helical peptide analog of ATSP 7041. So what I would say is they all work. They all work in terms of cell uptake, getting into the cytosol, and being able to provide you a readout using the nanoclick assay. Similar data has been achieved by Josh Quitcher using the kappa assay. So I guess we could say we have N equals 1 and 2. And so we can now use these assays as illustrated in the right hand of the slide in a yes-no format, which pharma likes to use originally, and now biotech as well, to answer questions. Do we have a potent enough binder? Is the peptide getting into the cell? Is the peptide active functionally in the cell? And if you're not functionally active in the cell, the question is why? Is it because you're not potent enough to bind to the target? Is your peptide degraded into metabolites that are not independently active against the target? Or is the peptide just not getting into the cytosol at all? We can now answer these questions with assays, including cell homogenate assay, which is a bit, simple and crude, so to speak, but it also is very helpful to at least know if your parent peptide is stable. So when we look in the context of cell uptake and intracellular targeting, I think now this slide here sort of sums it all up. We have passive transport in the case of cyclosporin A. We have lipid partitioning exemplified by ATSP 7041, stapled helical peptide. And we have cationic partitioning in the context of all kinds of CPPs, linear, cyclic, conjugates with payloads, hybrids that incorporate the CPP motif into the macrocyclic peptide. The peptides can come in all cases from experimental screening using libraries, phage, mRNA, DNA-encoded libraries, synthetic libraries. But my favorite, I would have to say, and certainly in the spirit of this conversation with you, Immanuel, is de novo design, utilizing the structural information of the target itself. So it's a new era that we're in. It's an era of knowledge. I'm not expert on AI, but I try to be expert on knowledge. So wherever those two things interface, that's where I kind of want to be. And if we're looking at simply hydrophobicity, which has always been a common denominator for many drugs in terms of permeability and intracellular targeting, as shown in the right-hand graph, and this is for analogs of ATSP 7041 that came out of the original series of work from Aileron working with Roche, we can see that the more you go to the bottom right of the graph, the more potent your peptides are based on the ratio of cell EC50 over target KD ratio. But if we look at experimental log D as the x-axis, where the most potent compounds are seen are also the ones that are most hydrophobic, that are having experimental log Ds in the 2.5 to 4 range. So the more hydrophobic, unfortunately, oftentimes the more insoluble. But in the case of the aileron compounds, the glutamic acid substitution rescued solubility rather significantly so that you could have both, hydrophobicity and solubility kind of in hand. I think that's an important challenge, and it can be accommodated by different ways, not just putting in a carboxy group. That's just maybe one way. If it works, great. It may not work all the time.
So first of all, when you're looking at the stapled peptide, the ATSP7041, and all the derivatives, when you were looking, and I guess you were comparing stapled versus non-stapled molecules, how massive was the effect of stapling on the cell penetration? That's first question. The second question: how soluble? So the end, like the ATSP 7041, how soluble was it in the end? I mean, orders of magnitude, just to get an understanding.
Yeah, good point. When we looked at linear analogs, I suggested that we just do 2 AIBs to replace the 2 staple amino acids to provide us with that helical bias. And so it was very interesting and a good question, because when we looked at MDM2, MDMX binding of the linears, they were actually awesome. In some cases, they were more tighter binders than the stapled helical peptide.
You're talking about a biochemical assay, not a cell
biochemical assay, yes.
That's actually surprising.
Well, the beauty of AIB, biasing towards the helix, has been known for a long time. And at risk of just picking one person to give credit to, I will anyway. Claudio Toniolo in Italy published a lot of beautiful work on AIB, looking at CD and other properties to try to make sense of it. And I think his work, and perhaps others during that same time, contributed a lot to the field. Unfortunately, it didn't seem that pharma biotech was jumping on helical secondary structure as their first hopes and dreams to make peptide drugs. And I think it's rather ironic that perhaps the 2 greatest marketed peptide drugs right now are the GLP-1s and insulin, each of which contain significant helicity, especially GLP-1. So I think that the AIB and alpha methylation kind of went to the wayside for quite some time and was only used here and there until, the work on GLP-1, especially from Novo Nordisk, pushed the field to think more seriously about the helix on receptor targets. And of course, aileron work based on the pioneering studies by Greg Verdine and Lauren Walensky and others. And now that has flourished way, way deep into beautiful chemistries. But it was interesting in the case of just going linear for ATSP, that for 7041, that the biochemical was really good, but the cellular was a disaster. There was no cellular activity. The peptide doesn't get into cells. I do believe that we made a azido-lysine modified version of the linear analog of ATSP 7041 at Merck. I'm struggling to remember if it's published or not, but it did support the fact that the peptide was not getting into cells. And so you needed that constraint. You needed that stabilized amphipathic helix in order for it to partition into the membrane and then translocate into the cytosol. That's my theory, that I think that evidence points to it significantly. And the other question, which was solubility, how much did that improve? A very good question, and my answer might maybe go beyond what you were asking. I think with peptides, the problem with solubility isn't just looking at the sequence and saying, oh, we got too many hydrophobics or too much grease, not enough hydrophilicity. It's a little bit more than that. It's also the ability of the peptides to aggregate with each other through backbone hydrogen bonding. And also you can get hydrophobic surfaces interacting as well to take away from solubility. But I think that when you introduce like N-methylation or macrocyclization, whether it's helical or what I call the donuts, circular macrocycles that are whatever, 7, 8, 9, 10, 11 amino acids or so, I think that gives you the ability to be very precise and with high fidelity make modifications that can improve solubility. It might be likewise to ATSP 7041, where we could introduce a carboxy in the hydrophilic valley of the amphipathic helix, or in the case of the circular or donut type macrocyclic peptides, it might be one or more side chains that may contribute to modifications that increase solubility. So I think that it's a bit still empirical. You can't get away from empirical. But the solubility improvement for ATSP 7041 relative to progenitors that did not have the glutamic acid was probably improvements of 10 to 100 times in solubility. So it was significant. Lastly, what I can say, especially from working with the Merck team, both US side, if you will, and Singapore, working with Anthony Partridge, in particular, my friend and counterpart there in Singapore, and David Lane, who is one of the fathers of P53, there's no doubt that intense biology, in vitro in particular, led us to publish this paper. We sort of picked on KRAS as the example to make some points, but fundamentally, I think it was a story that one needs to be very rigorous in your screening to eliminate the possibility of getting false positives. And in this case, we've identified 2 series of analogs that we found not to kind of make the cut, and actually were giving us false positive data. I think it's fair to say that in either case, modifications likely could overcome that. It was simply a issue with specific molecules. And in the case of the cyclorazins, Dewa has just published this past year or so an outstanding paper, which I highlighted earlier in the slide deck here, of a second generation KRAS inhibitor that would meet the criteria of certainly not being false positive, but being bona fide inhibitor. But I think it's important because nothing hurts the field more than, going after intracellular targets and then looking at simple functional readouts and perhaps not having all the tools to determine that you might be seeing off-target effects or so forth. So I think it's important to add rigor. So this paper was basically to bring it out to suggest tools that could be used for any investigators to ensure that their lead optimization is going in the way that they think it is. And then I would probably just wrap up this conversation, Immanuel, with kind of coming back to the waves and looking forward to our next one, which I believe will be very focused on AI and structure-enabled de novo design peptides, and maybe have the opportunity to hear maybe from you and the team with respect to what Pepticom is doing. I think that this is an exciting now new chapter in peptide drug discovery, and it could very well be the biggest chapter as we can now take the learnings of all prior work and experimental studies in particular and properties of peptides to achieve, the best molecules we can.
Sure. Thank you, Tomi. It was extremely interesting. And I think that intracellular targets are a very exciting field, though very, very challenging as well. And next time we will show you some ideas of how we approach peptide design using AI. And the emphasis is on design, meaning that we not only try to mimic nature by screening vast libraries of peptide, we actually try to design them with some ideas behind the design to make it actually interesting and functional and meaningful. In this regard, it's important to know that it has to be good enough to actually compete with strategies that screen 10 to the power of 12 molecules. So you need to have a very, very good framework in order to do that. And I think it's an exciting field, and stay tuned for the next time. So, Tomi, thank you very much for the talk. It was very, very interesting and eye-opening, and I think I for sure learned a lot. I hope the audience also were interested and learned, and looking forward for the next time.
All right, sounds good, Immanuel. So I'll say good luck to all my friends out there and best wishes to the new year. And we'll look forward to part 3 in 2023. See you, Emmanuel.
Happy New Year. Bye
All right,
bye.
bye.
See you
Bye.
soon.